Here I am again, Mr. Kleiman here. Today we're going to be looking at the details of DNA replication. So let's jump right into how DNA replication works. Last week we studied the history of this incredible molecule that's found inside of the nucleus of every cell of your body. It's an incredible history. But today what I want to do is fast forward to now and show you what we know about how exactly this molecule is copied and what exactly are the enzymes that help to do it. First thing that we need to address is why those copies are being made, and I'll remind you, of course, that relates to the cell cycle. For most of the life of any cell, they're living in interphase, this phase between cell divisions. Right after they divide, they go through a period of growth. Then during the S phase is when they actually do this copying of their DNA alongside their regular life processes of living their lives. And then right before they're ready to divide again, they go through a second period of growth. And of course, then they go into the phases of mitosis, or the splitting of the cell. And you're going to ultimately need two separate copies of that DNA to go into each of these cells. But those copies need to be 100% exactly perfect. Any error might make a protein stop working, and if a protein stops working, that cell is going to lose an important function. So if we have a look at this picture, which is summarizing the stages of mitosis, this is what we're used to looking at when we talk about DNA. You can see these little guys right here, these chromosomes. And if we zoom in, maybe here's my image of what a chromosome might look like. It's a single molecule of DNA with its associated proteins attached to it. When it's visible under the microscope during cell division, it's so coiled up, that it's actually inactive. We make that copy and it is attached to the original chromosome by what's called a centromere, and now those are called sister chromatids, and that little X thing you're looking at looks like this under the electron microscope. And as awesome as this molecule is, it's completely unusable in this form. This is so tightly packed and coiled, there is no direct access to the genetic code for either copying or for using. And so I want you to remember that if this right here is our chromosome, it's so twisted up and coiled and coiled and coiled that we need to unfold it before we can do anything else. So here you can see scaffolding protein, and then it's wrapped around these proteins called histones, but ultimately what we're trying to get at is that actual molecule itself, DNA, where we've got these complementary base pairs that we can unzip and then start to add complementary base pairs to make a new strand. So overall, this is what inactive DNA might look like, tightly packed and bundled. Here's that scaffold, and we're trying to access just that little molecule. The study of how DNA folds away and then unfolds for use is called epigenetics, and it's controlled by proteins that are on the outside of the DNA molecule. It's a fascinating area of research, and I really encourage you to have a look at it on your own time. So this is inactive, and this is DNA that's in use either for cell division or for making proteins. So let's just do a super quick review of the overall structure of DNA. I want you to remember that DNA is a polymer that's made out of the nucleotide monomers. Each nucleotide consists of a phosphate, a sugar, and a nitrogenous base, and we link those nucleotides via what we call the sugar-phosphate backbone. Phosphate to sugar to phosphate to sugar to phosphate to sugar, and the nitrogenous bases stick out to the side. This is the actual genetic code. I'll remind you as well the specific direction in which these things are linked. This right here is carbon number 5, and this right here on the neighboring sugar is carbon number 3. And so we call this the 5' carbon and the 3' carbon, and we always link them in this particular direction. This gives the molecule two different ends. Here's the 5' end, and here's the 3' end. When these nitrogenous bases sticking out to the side are linked to another nitrogenous base, its complement, A always links to T, and C always links to G. And you can see the difference in their hydrogen bonding pattern. 2 hydrogen bonds between A and T, 3 hydrogen bonds between C and G. But the only way for those hydrogen bonds to properly form is if the second strand of DNA is upside down relative to the first. We say that it is anti-parallel. If this is the 5' end of this molecule, then this must be the 3' end of the neighboring molecule, and there's the 5' end at the bottom over there. Once these two strands link together, this molecule is naturally going to start to coil up into this beautiful structure here that we call the double helix. So this is what we want to kind of unzip, and what I mean by unzip is break those hydrogen bonds so that we can gain access to individual strands of nucleotides so that we can make copies and read that code. And so how do we actually do that? Well, part of it is a bit self-powered. The energy that is going to be used in order to power this process is because we're going to use nucleoside triphosphates. And you already know one of those. It's ATP, but there's also CTP and TTP and GTP as well. And we can use the energy from these high-energy phosphate groups in order to power the process. Now this animation makes it look like it all does it on its own, but of course this is an enzyme-mediated process. And the main enzyme that's going to do this job can only add new nucleotides to the free 3' end of this sugar. And so that is going to be a really important theme, is that we can only work in the 5' to 3' direction. And so here's the overall picture of what we're going to try to do. Here's the parent strand of DNA. We're going to need to unzip it by breaking those hydrogen bonds. And then we're going to need to start adding new DNA nucleotides by complementary base pairing to these single strands that result. Those new strands are called the daughter strands, and by the end of that process we should end up with two exactly identical copies of the parent strand. Remember, this is the semi-conservative method of DNA replication. So how do we get in there? Well, if this is a coiled up molecule of DNA, there's going to be some starting point. We're going to call that an origin of replication. So I've simplified this molecule into these two red lines to represent two complementary strands. And so if this is the 5' end of one of those strands, this is the 5' end of the other. They're anti-parallel. And let's say at some point right here, there's a protein ready and waiting that's telling it this is the place to start to replicate. And so what's going to attach to that protein is our first enzyme to start the job, and that's called helicase, with the root word helix. It's going to actually be uncoiling the double helix of the molecule. And as it goes, it's going to move in two opposite directions. The result is that we get what's called a replication bubble. Now this begs the question, why doesn't this strand just reattach to the other through complementary base pairing? They're waiting to just re-zip, aren't they? So we're going to address some of these problems and challenges with some additional proteins in a minute, but for the time being, let's try to look at a simplified view of what's happening during DNA replication. Ultimately, what we want is not for it to reattach to itself. We want new DNA to be attached. So I'll be representing the parental strands in red, and I'll be representing the daughter strands in blue. And these are the copies that we need. And so at the ends of each one of our replication bubbles is what's called a replication fork. And so, of course, for each replication bubble, there must be two replication forks moving in opposite directions due to the action of helicase. In reality, there are multiple origins of replication around DNA because the molecule is so enormous that we need to get this process moving a bit faster. So we can start from multiple different places, and each replication bubble eventually will meet up with the other via these replication forks, until we have two complete daughter strand molecules. But there's one last critical problem that is going to happen in this region right here as two replication forks are unzipping that DNA towards each other. And it's the same problem you might imagine if you've got this rubber band and we're trying to uncoil it from both ends. We're going to develop tension in that coil in between those two replication forks. And so we've got two enzymes that are going to help us deal, or two proteins that are going to help us deal with these problems. So here's our trusty helicase unzipping the DNA molecule, and our first helper is the single-stranded binding proteins that prevent the DNA from complementary base pairing back with itself okay, and reforming that double helix. This is going to temporarily hold that DNA as two single strands. But working upstream of helicase, ahead of it, is another enzyme called topoisomerase. And what it does is it spins and coils around that double helix far ahead of helicase. And when there's enough tension, it's going to grab onto that DNA molecule in two places, cut it, let it uncoil to relieve that tension, reattach, and then keep going. And so now we've dealt with those problems of the replication bubble. We're ready to make some copies. So if this is our replication bubble, now we can start to copy. But there's no starting point yet. The main machinery for making a copy, for adding DNA nucleotides to this already existing DNA strand, requires a starting point. It needs a free 3' end of an already existing nucleic acid in order to start adding new nucleotides. And so the first enzyme that comes in is called RNA primase. And what it's going to do is add a temporary little binding primer, which has a sequence that's going to recognize some sequence on the DNA, and it's going to attach a little piece of RNA. And now, if you look at that RNA primer, which is just a little segment of RNA bound to this DNA molecule through complementary base pairing, okay, it's anti-parallel to that parent strand. So if this is the 3' end of the parent strand, this must be the 3' end of that RNA primer. This is the free spot where the next enzyme can start to work. And that next enzyme is called DNA Polymerase III. And yes, there is a DNA Polymerase I and II, and we're going to revisit those in a minute. But DNA Polymerase III is the main workhorse of the process of DNA replication. And it works in only one direction. It must work in the 5' to 3' direction. In other words, it requires a free 3' end, and it can only keep adding nucleotides to that side. And so here they go in opposite directions from those primers, working towards the replication fork. And that's awesome. We're going to make a perfect copy using RNA Polymerase III. But we're going to call these the leading strands because they've created a bit of a problem. While they're working towards the replication fork in this direction, behind them they're leaving this gap. As these helicases continue to work in opposite directions, how are we going to fill in these ever-increasing gaps? We can't add DNA nucleotides to the free 5' end. And so the solution is we're going to need some more enzymes. So while the leading strand always grows towards the replication fork, now we need to deal with this gap on the other side. Back comes RNA primase to add a new primer. And now we have a new free 3' end, and we can continue to work in the 5' to 3' direction to build little fragments of DNA. And so as we go, we're going to start to create what's called the lagging strand, which is growing away from the replication forks. So the lagging strand is going to need to be built in little segments. Now, as you can see, we have a couple more problems to deal with. The last nucleotide we've added needs to be stitched on to the already existing strand. But this pink region here represents not DNA, but it represents RNA. So we need to replace the primers with DNA, and then we need to stitch together the sugar phosphate backbone of each of these fragments of DNA. And so here's where our last few enzymes come into play. DNA polymerase I is going to replace RNA nucleotides with DNA nucleotides. There it goes. And the last enzyme we need is called DNA ligase, which is going to link the sugar phosphate backbone of the adjacent pieces. And there we go. There's our copy. So here's our replication bubble. Leading strand working towards the fork, lagging strand away. Getting bigger, the leading strand continues towards the replication forks, and the lagging strands get built in smaller fragments. And we call those little fragments Okazaki fragments, named after the Japanese scientist who discovered them. And so here's a nice little summary diagram of all of those components we just saw. Here you can see DNA helicase opening up a replication fork, and in complex with it is a primase, which is adding our primer molecules to it. Here you can see our single-stranded binding proteins holding it out as a single strand. There's our little piece of RNA primer that's been attached, and here you can see a DNA polymerase working from that primer away from the replication fork. This is, of course, the lagging strand. The other strand, as it opens, we at some point way back here have added a primer, and DNA polymerase III just keeps on working towards that replication fork as helicase works in this direction. No new primer is needed. Retrieval practice time. Try to name each one of these enzymes and record what each one does. In reality, all of these enzymes that I'm describing to you are working together in this beautifully orchestrated dance. And in fact, each one of these enzymes is grouped together into this unbelievable complex where all of the enzymes are working together. Here you can see DNA polymerase III on the leading strand. Here you can see it on the lagging strand. And what happens in reality is that these guys are all in complex together and working simultaneously in order to create a strand. So let's take a minute to watch a 3D animation of what this actually might look like if we could see it with our eyes.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
That's all I have for you today, folks. I wish you the best of luck. This is one of 3 videos that I'm going to produce today. Our next topic is to look at how we now take DNA and turn it into proteins. So enjoy.